Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about amino acids and proteins. Proteins are polymers or macromolecules, and amino acids are their building blocks and micromolecules. So let's start. So at first it is the building block of proteins, all 20 standard amino acids. Standard amino acids which takes part in the protein formation, okay, during translation process, and non-standard amino acids are manufactured during any biochemical pathway, which are the byproducts of any biochemical pathway. Those non-standard amino acids are not directly prepared by translation process. So the standard amino acids share a central alpha carbon bound to hydrogen atom, amino group, carboxyl group, and a variable side chain. Okay, to this side chain any functional group can be attached. Okay, next, special side chain. So some amino acids have special R group or side chain, like glycine. Here the side chain is a single hydrogen atom. That means, it is the alpha carbon, one hydrogen is there, COOH group, NH3, and here in place of side chain hydrogen atom is there. It is the structure of glycine. It is the most simplest and smallest amino acid. Next, fits into tight spaces and adds high flexibility to protein chain. So it is the case of glycine. Proline, the ring forming constant. The side chain bonds back to its own backbone nitrogen, and it forms rigid ring structure actually, breaks the alpha helices by introducing sharp kinks. So it forms a ring. Next, cysteine, the disulfide marker, contains reactive thiol or SH group. It forms covalent disulfide bridges with other cysteines. It stabilizes and locks the protein shape. Next, histidine, the features an imidazole ring side chain, has pKa very close to body pH, and imidazole ring structure, which is a 5-membered aromatic ring with 2 nitrogen atom. Okay, and pKa value is roughly 6.5 to 7, which matches the normal body fluid, pH of normal body fluid. Okay, so what is pH and pKa? pH is the measure of how acidic or basic a liquid solution is, based on the concentration of its hydrogen ion it contains. But pKa is fixed chemical property of specific molecule, which tells you about how easily it gives off hydrogen ion. So that is the difference between pH and pKa. Okay, next, standard and non-standard. I have already told you standard amino acids involved in the protein formation, but non-standard amino acids are not directly coded by DNA during translation. Instead, they arise or manufactured from any biochemical process, chemical modifications during post-translational modifications, after protein synthesis, or alternate biological roles. So these are difference between standard and non-standard amino acids. Another thing you have to remember about acidic and basic amino acid. Acidic and basic amino acids. Acidic amino acids which are negatively charged, and basic amino acids which are positively charged. The examples are aspartate, glutamate, and basic amino acids are lysine, arginine. So it is the case of acidic amino acid. They have side chains with extra carboxyl group. So they have extra carboxyl group in their side chain which can lose protons. Okay, and basic amino acids have extra amino group or nitrogen rich side chain that accept proton and becomes positively charged. So these are the case of acidic and basic amino acids. Now the chirality of amino acids. 19 of 20 common amino acids are chiral because they are central alpha carbon bonds to 4 different groups: amino group, carboxyl group, hydrogen atom and a variable side chain. They form non-superimposable mirror image pair called enantiomers. The amino acid can have L form and D form. Exception is glycine, which is achiral in nature, because glycine has a hydrogen atom as its side chain and its alpha carbon bonds to 2 identical hydrogen atoms. So no mirror pairs is there. It lacks 4 distinct groups, so it cannot form optical isomers. It lacks 4 distinct groups because it have hydrogen atom along with the existing hydrogen atom as its side chain. So it lacks symmetry. Okay, next, D and L configuration. So any amino acid can have L form. Most biological proteins use L form amino acids, and D amino acids appear rarely, such as in bacterial cell wall. In bacterial cell wall, D amino acids are present. D amino acids. So what is L and D amino acids? So these are the chirality. Except glycine, the central alpha carbon binds to 4 different groups. This creates non-superimposable mirror images. In case of L configuration, in a Fischer projection, what is Fischer projection? It is the 2D shape or 2D picture of a 3D structured amino acid. Okay, and L configuration, here in case of L configuration, in a Fischer projection with carboxyl acid group on the top, the amino group or NH2 or NH3 group points to the left. But in case of D configuration, the same projection layout, the amino group points to be right in case of D amino acids. Okay, so like this. Next, what is isoelectric point? Isoelectric point of amino acid is specific pH value where the molecule carries no overall electric charge. At this pH, the amino acid exists as Zwitter ion, meaning its positive and negative charges completely balance out. That is the case of isoelectric point, where the molecule or amino acid molecule carries no overall electric charge. Okay. Now, peptide bond. The peptide bond is formed during the protein formation, during protein manufacturing. So peptide bond is formed between 2 amino acids, the carboxyl group of one amino acid to the amino group of the next amino acid. During this peptide bond formation, one water molecule is removed. Okay, and finally peptide bond is formed. Now about the Ramachandran plot. Actually, it should be studied after protein structure, but I will tell you now. So Ramachandran plot is fundamental tool in structural biology used to visualize or imagine energetically allowed regions for backbone dihedral angles in a protein. Okay, so allowed region, it is a graph. Actually, it is a graph that shows the allowed and not allowed angles for backbone of a protein during protein structure formation. It shows whether it is allowed region or not allowed region. Okay, This plot first created in 1963 by G. N. Ramachandran. So according to his name, this plot is named as Ramachandran plot. It maps phi and psi dihedral angles. Dihedral angles, what is dihedral angles? So this angle is geometric angle between two intersecting planes, or half planes. It is measured on third plane that sits at right angle to the line where first two plane meets. It is the, suppose it is one plane, it is another plane, it is the dihedral angle. Okay, so it is about Ramachandran plot. Next, peptide bond formation during protein synthesis or protein structure formation. So when amino acids join with each other to form the primary or linear structure of protein, they form peptide bonds between them. Peptide bond is a strong covalent bond. Actually, peptide bond and disulfide bond both are strong and strong covalent bonds. So peptide bond links amino acid into chain, while other hydrogen bond, ionic bond, disulfide bond, hydrophobic interactions fold and stabilize the protein into its final functional state. So they said that peptide bonds actually link two amino acids during the protein linear chain formation. Once the primary structure of protein is formed, then hydrogen bond, ionic bond, disulfide bond and other hydrophobic interaction fold that linear chain of amino acid and stabilize protein into its final functional shape. Next is disulfide bond. And about peptide bond, I have already told you about peptide bond formation. It is formed between the carboxyl end of one amino acid to the amino group of the next amino acid, and during the formation of peptide bond, one water molecule is removed and finally peptide bond is formed. Now disulfide bond, it is also a strong covalent sulfur-sulfur link formed between two cysteine side chains that stabilizes the folded structure of protein. Next, stabilizing non-covalent interaction. First one is hydrogen bond, next ionic bond, different types of hydrophobic interaction, van der Waals forces. These interactions actually help in the folding and stabilizing of protein structures. Next, structure of protein. So there are four levels of structure. Primary structure, which is the linear sequence of amino acids. Secondary, which include local folds like alpha helices and beta sheets. Tertiary, which include overall 3D shape of single chain. Quaternary, multiple chains working together. So now it is the primary structure of protein, which is formed by linking the amino acids by peptide bond. Okay. Next, secondary structure. So secondary structure of proteins is local repeating 3D shape of polypeptide chain. It forms by the hydrogen bond between carbonyl oxygen and amide hydrogen atoms of the protein backbone. There are two main types of secondary structure. One is alpha helix, which is right-handed coil or fine spiral shape of protein stabilized by hydrogen bond between carbonyl oxygen and amide hydrogen. Each turn contains 3.6 amino acids. It provides flexibility and elasticity. Next is beta pleated sheet. Beta pleated sheet is another form of secondary structure of amino acid. It is made up of stretched polypeptide strands. called beta strand formed by hydrogen bonds between adjacent strands. Strands can run in the same direction or it can run in opposite direction. When it runs in same direction, it is called as parallel. When it runs in opposite direction, called antiparallel. This type of beta pleated sheet structure or secondary structure provides strength and rigidity to the protein. Okay. Now the tertiary structure of protein. It is overall three-dimensional shape of protein. It forms when secondary structure like alpha helices and beta sheets fold again. This folding happens because amino acid side chain interact with each other and with water. So what are the main key forces or stabilizing bond? Hydrophobic interaction, hydrogen bond, ionic bond, disulfide bond, van der Waals forces. These forces stabilize the bonds present in tertiary structure of proteins. Now the quaternary structure of protein. Here it is the arrangement of two or more separate folded protein chains or subunits into single larger functional complex. Not all proteins have this label. It only occurs in protein made up of multiple polypeptide subunits. For doing a function, a typical function, most common example is hemoglobin made up of 4 subunits, DNA polymerase, many antibodies, those have quaternary structure of proteins. Okay. So these are different labels of protein structures. Now what is motif and domain? So protein motif, it is a small recurring super secondary arrangement of secondary structure that cannot fold or exist independently, and it is simple combination of few secondary elements, and it loses its shape and stability if separated from rest of the protein chain. But protein domain is larger, compact, stable 3D shape structural unit that can fold independently and often carries out a specific function. It is large, comparatively stable than motif, functional module made up of multiple secondary structure and motifs. And domain fold properly and remain stable even if isolated from rest of the proteins. So this is the key difference between motif and domain of protein. Okay. So with this I am concluding my lecture video here, and I hope I have covered important chapters, important points of amino acids and proteins. In my next video I will discuss another important chapter of your CSIR NET Life Science Biology syllabus. So please stay tuned and subscribe to my channel Sunanda's Tutorial. Thank you.